Meiosis. In this video series, you are going to learn how things are inherited, how there's variation when things are inherited, how do we get new genetic information. Okay, so let's go through Meiosis 1. Meiosis 1 has 4 separate stages, or we call those phases. I like to call them PMAT. So the first one is prophase. In prophase 1, the nuclear membrane breaks down. It's represented by this dotted line right here. That's the nuclear membrane, the membrane that surrounds the chromosomes. It starts to break down. And these centrioles, you remember from cell organelles, separate and make these spindle fibers. These homologous chromosomes are going to pair up and they'll become visible. So these are homologous pairs, homologous chromosomes. They're duplicated chromosomes, and they are going to form these tetrads. These are a cluster of these 4 chromatids. 1, 2, 3, 4. And this all together is what we call a tetrad. Now when this happens, they start to get really close together, and that's when crossing over can happen. If you remember from the other diagram how it looked like one piece of DNA from this chromosome ended up on like another chromosome, that's crossing over. Crossing over occurs between homologous pairs, homologous chromosomes, because when the chromosomes get really close together, it recognizes that that's the same genetic coding for eye color. One might be blue and one might be brown, but it's the same genetic coding, that's the same gene. And sometimes they will just switch. Crossing over occurs during prophase 1, and when the homologous pairs are lined up all together, these chromosomes can get sort of like tangled. It's the best way I have to describe. And when this happens, they'll just swap those pieces of DNA. And this process happens because we create new combinations of genes. And these chromosomes are now part mom and part dad on this chromosome, if you will. But it's also kind of mixing it up a little bit so that now not every single one is the same. This is how we get genetic variation among families, even among species. The second part, metaphase 1, these homologous pairs, they're going to line up in the middle of the cell, metaphase, lining up in the middle. And these centrioles have created these spindle fibers, and they're going to attach to the individual chromatids, and the cell's going to get ready to divide. Metaphase is the most recognizable phase, I feel like, because you will see cells and they'll have all of their chromosomes, like lined up in the middle of it. And middle, that's an identifiable thing in a real cell and in a drawing or illustration. Now in anaphase, these homologous chromosomes are going to separate. Each side is going to have a pair of these sister chromatids. That's a sister chromatid, that's a sister chromatid, and they're going to get pulled away from one side of the cell. Now these sister chromatids are going to remain attached to each other in anaphase 1. But in anaphase 1, usually these spindle fibers and centrioles, they're still visible, and you can see them migrating to the opposite sides. Rounding out the end of our PMAT, we're now at the T, telophase 1. This is where chromosomes are going to gather at the poles, like one pole or the sides of the cell, and the spindle fibers and the centrioles are going to eventually disappear here, but the nuclear membrane is going to start to reform. It was always there before, it just kind of went away or broke down in pieces so that the cell could replicate. So this nuclear membrane is reforming, and telophase is easier to see in a drawing because you can identify with things being at these poles like this. cytokinesis, or the splitting of the cytoplasm, the cell has to divvy up all of its nutrients and goodiness that's inside of it, not just its DNA, and contribute some part to each of the new cells. And so cytokinesis is when the cell divides into two, splitting its cytoplasm. And the end result is going to be these two haploid daughter cells with duplicated chromosomes. And they're different from the sets in the original diploid cell. Now we're going to start meiosis 2, and we have these two daughter cells, and again, prophase 2, the nuclear membrane is going to break down, and these spindle fibers, they're going to come out and attach to those centromeres. Centromeres are the center parts of the chromosomes right here, and we're going to divide up these sister chromatids. Sister chromatids, they're going to line up in the middle, once again, metaphase 2. And these sister chromatids, when they line up in the middle, it's kind of like that single file. But in metaphase 2, we have two cells right here, and these centrioles are out, that is spindle fibers. They're attaching to the centromere on that chromosome and the centromere on the other sister chromatid. And in PMAT part 2 here, anaphase 2, these sister chromatids are going to separate. So the separating of sister chromatids is in anaphase 2, and you can see how they're being pulled apart to either side of the poles away from each other. And anaphase 2, it looks like we're about to have 4 different cells, because if you look, like, you know, you can see 1, 2, 3, 4. It's more defining that we're going to get to that 4 gamete cell structure here. And so finally here, telophase 2, that's when the nuclear membrane starts to form. Now, I suppose I should mention this nuclear membrane is going to form before the cell completely divides its cytoplasm and splits up. I always felt like it was doing this because we're just going to make sure that we separate the DNA first, because we don't want that getting mixed up. We don't want DNA from over here to kind of get over there, because remember, this is more of a fluid-like structure, cells are, and we don't want errors happening. So the nuclear membrane starts to form in telophase 2, and these spindle fibers will start to disappear, and they'll just kind of dissolve back into the cell. And then in cytokinesis will happen again. Cytokinesis is just this division of the cytoplasm, and it will split up into 2 individual cells. And so from the original 2, we now have 1, 2, 3, 4, and these are called haploid. Remember, haploid daughter cells. And each of these cells, or these gametes, let's just say they could potentially be 4 different sperm cells. They contain half of the genetic information for a new baby. So they're only N, right? Their DNA is only N. It only has half of it. And so the end result of meiosis, the creation of these gamete cells, is so that sexual reproduction can occur when it meets up with the other N to form a 2N cell. But essentially, one cell in gametes inside of your gonads has produced this genetically unique individual sex cell with combinations of all of your DNA and ancestral DNA, and each one of these sperm cells can produce a different looking, different phenotype baby.